Welcome students to this module of e-learning. I'm Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present to this module to the first year students of Botany, Semester 2, Course Code BOC 102 and the title Biodiversity 2, Vascular Plants. Paper titled Biodiversity 2, Vascular Plants. The module name Taxonomic Evidence from Palynology, which belongs to Unit 5, Taxonomic Evidances from Palynology, Cytology, Photochemistry and Molecular Data. The outline of the module will be Definition of Palynology, Basic Evidentiary Characters of Pollen Grain, Examples of Taxonomic Evidences from Palynology. Once the student completes this module, the student will be able to explain the terms like taxonomic characters and palynology, describe the evidentiary characters of pollen grains, and the taxonomic evidences based on palynology. Dear students, what does taxonomic character mean? They are the attributes that can be used to provide evidences from which relationships between taxa can be inferred. Now, if we see the word palynology, it is dealing with the study of pollen grains, and it comes from the Greek verb palyn, means to scatter. Now, the pollen grains are disseminating aid by different agents of pollination, and they are found in every nook and corner. Palynological evidences appear very effective, students, in plant taxonomy and phylogeny, since pollen grains possess distinctly unique characters which are genetically controlled. Pollen grain morphology plays an important role in classification. Study of pollen grains has been proved to be very useful in solving taxonomic disputes. Now, there are several pollen features useful in taxonomic delimitation, like pollen unit type, pollen grain polarity, exine structure, exine sculpture, aperture, shape, type, position. There are a number of them to list. If you see the pollen aggregation or pollen unit type, we see monads, which are seen in majority of the angiosperms. Dyads, which are seen in the rare cases like Magnoliidae. Trinucleate are seen in Caryophyllaceae. Tetrad also are seen in number of plants, which we'll see in the next slide. Polyads are seen in Calyandra. Maculae, they form irregular groups in Orchidaceae. Whereas Pollinia, they are seen very much in Asclepiadaceae. Now, the tetrad also are seen in particular families like tetrahedral tetrad in Ericaceae, linear tetrad in Typha, rhomboidal tetrad in one plane with two separated from one another by close contact of the other two. This is the one, students. Tetragonal tetrad in one plane seen in Philodendron, and decussate, that means in two pairs. That means they arrange at right angles to one another, seen in Lactanthus. We also see a lot of variation in pollen sculpturing, students. We see baculate, clavate, foveolate, reticulate, fossulate, vericulate, gemate, psilate, striate. Specifically in the genus Taraxacum, we see echinate type of pollen grains, and they are having spines less than 1 microns. Whereas in Quercus species, we have scabrate pollen grains with spine-like elements which are less than 1 micron. I did a mistake here. We also see, students, a large number of pollen wall sculpturing types. We see, I have to erase this 1 minute. This is greater than, and I said less than. Okay. We see a lot of variation, students, in the pollen wall sculpturing. We see baculate, clavate, foveolate, reticulate, fossulate, verrucate, gemate, ciliate, striate, etc. Particularly in the genus Tarasaxum species, we see a kinnate type of pollen grains, wherein the spine-like elements are greater than 1 micron. Whereas in Quercus species, we see a scabrate type. And in this, the spine-like elements are less than 1 micron. There are a number of variations with regard to the pollen aperture as well. We have 2 types initially: inaperturate, that means there is no aperture, and aperturate, that means there's a presence of an aperture. The aperture can be porate, colpate, sulcate, if it's a pore, it's a furrow, or a sac-like structures. It can be mono, tri, porate, colpate, or sulcate. Now, depending upon the palynological evidences, 2 distinct phylogenetic stocks have been seen in the dicots, have been suggested. One is Magnoliaceae and Ranunculaceae, wherein Magnoliaceae shows monocolpate pollen grains, that means a single colpi, whereas tricolpate means it's having 3 colpi. Now, if we see the angiosperms, the monocolpate conditions is widely distributed in primitive dicots and a majority of the monocots. And the monocots are considered to be closely related to the Magnolian stock on the basis of monocolpate pollen grains. The pollen grains of Anemophilus plant, the one which are being disseminated by wind, they are usually small, round, smooth, thin-walled, dry, seen in the families Poaceae, Cyperaceae. They can be easily disseminated, students, because of their structure like this. Whereas the ones which are disseminated by insects, birds, on the other hand, they are large sculptured, often coated with adhesive, waxy or oily substance. Now let's see step by step at the family, genus and species level how palynology has helped in solving taxonomic disputes. On the basis of palynological character, with regard to family level, Fumaraceae is separated from Papaveraceae and Nilomberaceae from Nymphaceae. Now if we see Nilombo is a genus which shows consistently tricolpate pollen grains. Now, it was originally placed in the family Nymphaceae. Nymphaceae family shows monosulcate pollen grains. Now, because Nilombo shows tricolpate pollen grains, it is shifted from Nymphaceae and placed into a separate family, students, which is Nilombonaceae. So, this is at the family level. There are some families also, students, which are recognized on the basis of pollen sculpture. Malvaceae and Bombycaceae are separated on the basis of exine characters, wherein Malvaceae shows spinous exine, whereas Bombycaceae shows reticulate exine in pollen grain. In addition, Malvaceae and Asteraceae have both spinulose exine. Plumbaginaceae has verrucate exine, Poaceae has smooth sulcate exine, Nepenthaceae and Drosseraceae, except Drosophilum, has spinulophorus pollen tetrad. Such type of pollen tetrads are not found in any other plant. Relationship between Polygalaceae and Ephidraceae are based on similarity between their pollen grains. Now we'll see the family Beberidaceae. The family Beberidaceae consists of 12 genera. So modern taxonomists, what they did is they removed the genus Podophyllum from this family Beberidaceae and placed in a separate family Podophyllaceae, because on the basis of pollen aggregation. Because Podophyllum, the pollen grains remain united. The pollen grains in Podophyllum remain united, but they are free in other genera. This shows the role of palynology in taxonomic delimitation at the family level. At the generic level, the role of palynology is delimitation at genus level is evident from the family Salicaceae. Now, Salicaceae family has 2 genera, Salix and Populus, and these are distinguished based on the pollen characteristics. The genus Populus has spherical pollen grains without distinct apertures, whereas Salix is long, narrowed and 3-furrowed pollen. There are seven genera of Polygonaceae, that is Coninia, Persicaria, Polygonium, Pleuropteropyrum, Bistoria, Tininaria and Phagopyrum, and they are different all in their pollen morphology. In Phytolaceae, the pollen of Phytolacca is 3-zonocolpate, whereas in Rivina, it is pantocolpate. The pollen characteristic helps in differentiating species within a single genus. Now last, at the specific level, we have the genus Anemone. Buttercup belongs to Ranunculaceae. Now this genus, there are 4 species in this genus: Obtusifolia, Aschelemain foliate, Rivularis and Fulgon. Now these 4 are distinguished. These 4 species of anemone are distinguished based on their pollen grains. Obtusifolia shows 3-zonocolpate. The second species shows pentaporate. Third one pentacolpate, and Fulgon shows spira-aperturate. So at the specific level, based on the exine pattern, different species of Bauhinia fabaceae can be differentiated. Now see students, pollen size played a very vital role in demarcating different species of Malva belonging to Malvaceae. So you see how palynological evidence have helped at the family level, generic level and the species level. So students, these are the references. These are the online references. And thank you students for a patient hearing.